All rise, please. Please be seated.
I hereby open this academic ceremony in which Alexander Kroner will defend the academic thesis, A Deeper Look at Spatial Attention, the Computational and Neural Basis of Visual Saliency. May I invite you to present a summary of your study and the conclusions of your thesis?
Highly esteemed Pro-Rector and members of the opposition, dear supervisors, colleagues, friends, and family. Thank you all for coming to my defense, either in person or online. In the next 15 minutes, I want to summarize the work that is part of my thesis, A Deeper Look at Spatial Attention, the Computational and Neural Basis of Visual Saliency. Now, during my PhD, I've studied the topic of saliency in the visual domain. And to get started, I first want to introduce you to the concept of saliency, to explain what it is, why it's relevant, and also how it can be studied so that we can get a better understanding of saliency. Now, when we look at complex natural images like the one here, then the way we move our eyes across this image is not random, but instead it's highly strategic. So our eye movements are fairly consistent across images like this, and also there's a very considerable overlap between observers that look at the same image. So chances are, if you look at this image here, that you focus on the street sign, because that street sign carries a lot of meaning for the context that the street sign is in. Now, if we want to understand where people look at exactly, we can measure the fixations via eye tracking. So here you see the fixations accumulated over different observers of the same image, and all of these individual fixation points then. And indeed, many of these fixations land on the street sign. Now, eye tracking is not perfectly accurate, so to account for these inaccuracies, what's often done is to then blur these individual fixation points, which gives us this distribution or a heat map over the scene. And this distribution is commonly called a saliency map, because what it does is that it indicates which parts of the image are likely to attract our attention. Now, if we want to understand what saliency is, we can do so from two perspectives. One is a computational perspective, the other one is a neuroscientific perspective. The computational perspective addresses questions like, what are the features that underlie where we look at in a scene? And how can we then exploit this knowledge about these features to build predictive models of saliency? On the other side, the neuroscientific basis asks questions like, is there anything like a saliency map also in the brain? And what are the regions that are involved in saliency processing? Now, over the course of 3 chapters, we address the topic of saliency from these two perspectives. Chapter 2 looks into developing a computational model that makes predictions of saliency based on natural images. Chapter 3 then relates computational models to the brain to understand better where saliency may be represented in the brain. And in Chapter 4, we look at a region that is heavily involved in attentional processing to better understand the spatial organization of this region. Now, let's get started with Chapter 2, where the goal is to map natural images to a saliency map. So the idea is to make predictions that mean where would someone fixate if they were presented with such a scene. Now, in general, visual information can be broken down into simpler parts. And these simple parts are properties of an image or so-called features. Now, there are simple features that we can break images down into, like luminance information, color information, or as you can see here, oriented edges. And based on our knowledge of these low-level features that occur in an image, we can make predictions of saliency that look at the contrast, so at luminance contrast, color contrast, or orientation contrast. And a prediction based on these low-level features would look like this. Now, to better understand if it's a good prediction of saliency, we can then compare it to measured fixation patterns in a scene. And as you can tell, there's not a great overlap here between the predictions of saliency and the measured fixation patterns. And that just shows us that low-level features are just not highly predictive of where someone would look at in a scene. So what we can instead do then is to represent images according to more complex features. So we can combine low-level features into mid-level features that you can see here in the middle. And these mid-level features can be then combined into entire object-based representations that you see here on the right. And then based on these object-based representations, we can again make predictions of saliency in a scene. And that would look like this. So that's a high-level account of saliency. Now we can again compare it to where people look at indeed, and there's a much better overlap in this case now. So what we can conclude from this is that high-level features are just much more predictive of where someone fixates in a scene compared to low-level features. Now, one thing that we did here is just to define these individual features by hand. But that's not a very scalable solution. It's not feasible to do all of this by hand. So we need a better way to do this. And that's where machine learning comes in very handy, because instead of defining all these features by hand, we can extract them and we can find them automatically. And that's what a model class called convolutional neural networks does. It represents features according to so-called feature maps. And these feature maps can be combined into layers. So what you see here is 3 layers that each contain 3 feature maps. And then these features can be detected based on a learning task. So we can take these individual building blocks, these layers, and we can stack them and scale them up. That gives us an entire network architecture, as you see here, that consists of many layers that extract increasingly complex features from a natural image. And such a network then has to be trained on a specific task. And one such classical example is image classification. So what happens here is that a natural image is shown to this network. It goes through all these processing layers where features are extracted. And at the end of it, it gives an output which represents the object that is present in a scene. In this case, a skyscraper. Now, the features that are relevant for performing an image classification task can also be repurposed for other kinds of visual tasks, such as saliency prediction. And that's what we did. We used this feature basis from a classification network, and we used it for building a new saliency model. And this saliency model has an encoder which extracts these features of increasing complexity, and it has a decoder that takes these feature representations and transforms them into a prediction of saliency. Now, this is an approach that has already been done before. We were not the first, and one addition we do here is the so-called ASPP module in the middle of the network. Now, what this model does, and it stands for Attrue Spatial Pyramid Pooling, is that it doesn't just look at a single spatial scale when it comes to detecting features, but in fact, it integrates multiple spatial scales to make a prediction. And it also computes something called the scene gist. So that's a summary, a very coarse summary of what is present in the scene. And this scene gist has also shown to influence where we look at in an image. So that's why we introduce a module that captures these 2 aspects. And then when we build our entire model with the encoder, this ASPP module, and the decoder, we can make predictions again. And here you see 2 examples where our model predictions align very well with the measured fixations from humans. And that also generalizes to many other images. So that really shows that our network is very capable of making these sorts of predictions on natural images that it has never seen before, in fact. Now, building on top of these computational models that we built, we can then relate computational models to the brain. And that's what we addressed in chapter 3, where we wanted to investigate which regions are highly relevant for saliency processing. Now, in general, many regions have been suggested to play a role when it comes to saliency and possibly even house an explicit saliency map. All of these regions, or some of these regions that were mentioned, are listed here. Some of them are early on during the processing hierarchy, like LGN and V1. And then information splits up into the dorsal and the ventral visual stream. Now, the dorsal visual stream has most prominently been mentioned in the context of saliency processing, especially area IPS, but also areas that are more linked to eye movements, such as FEF and superior colliculus. Now, more recently, the ventral visual stream was also mentioned when it came to saliency and attention processing. And among those, V4 and IT, those regions. And those regions may be more involved in a high-level account of saliency that considers these object-based representations rather than only low-level features. And what you can see here is that there's no real consensus among which regions are involved in saliency processing, if there's a single region that houses a saliency map, and how we can combine all these information. So for that reason, we wanted to revisit this topic and look at it from a more computational angle. And for that, we also need neural data. So luckily, there's a data set called the natural scenes data set, which contains many neural responses to natural scenes being shown to 8 participants. And these neural responses are measured via functional magnetic resonance imaging, or fMRI for short. Now that we have our models and some data, we can then come back to the two sort of opposing questions. Is there a single saliency map in the brain, or is it possibly a more distributed account of saliency processing? So the approach we take here looks like the following. We do many model comparisons, but I want to focus on one here. So the images that were shown to the participants in the natural scenes dataset were also shown to 2 computational models. One is a classification model, and the other one is a saliency prediction model. And by feeding natural images to these 2 models, we can extract feature activation from these computational models. And based on these feature activations, we can make neural predictions. And looking then at our neural predictions, we can then infer which of the 2 models is more capable of explaining neural data. And here are some of the results we got. So what you see in red is regions that are better predicted by a classification model, and areas in blue that are better predicted by a saliency model. And all the areas that are listed here are the ones that are better predicted by the saliency model. And what you can see is that there's quite a distributed pattern. There's not a single region that is better predicted by saliency compared to the classification model, but it's many regions at that. And so we think that it's probably the interplay between all of these regions that gives rise to where we look at in a scene when it comes to natural images. Now in chapter 4, we then narrowed down on a region in the dorsal visual stream that has been suggested to play a very important role in attentional processing. And we wanted to address the spatial organizational principles in this region by means of comparing different experimental procedures. In general, if we want to map areas in the visual system, then what's typically done is that a certain stimulus is presented very systematically across the visual field. And here you see one such example, where a flickering checkerboard bar is presented at different positions in our visual field. And then based on this presentation, a neural activity is elicited that can then again be measured, for example, via fMRI. Here we look at an example where activity in the early visual cortex is recorded. Now, with our knowledge on where a stimulus was presented and the corresponding neural activity, we can build a so-called population receptive field model. So what this does is that it looks at a certain neural population in our brain, in this case in the early visual cortex, and it tries to characterize the preferred location in our visual field that this neural population responds to. So in this case, we have a population in the left hemisphere, and this left hemispheric population responds to a location in the upper right quadrant of our visual field. Now, this is a receptive field then, so the preferred location in our visual field. And this receptive field can be characterized according to 2 measures. One is the eccentricity, that's the distance from the image center, and the other factor is the polar angle, so at what angle this receptive field is located. Now, taking into account these 2 measures, what we can do is to look at preferences across the early visual cortex. So here you see that for both eccentricity and polar angle measures, you see gradual changes in preference across the early visual cortex. Now the question is, can we find similar organizational principles also in a region in the dorsal visual stream that are more implicated in attentional processing? And for that, we look at different types of experiments. Here you see one such example where a bar is presented at a certain location in our visual field, and this bar contains 3 segments, and each of these segments has a certain motion direction. And the task for the participant is to then indicate which of the 2 outer segments matches the motion direction of the inner segment. And that's an attentionally very demanding task because this bar is not presented for very long because we want to really have the participant pay close attention to the stimulus. And we didn't only use this experiment, but also 5 other ones that all vary in a way of how they elicit attention. But the goal in all of these experiments is that people have to pay very close attention to the stimulus. And now, based on these experiments, we wanted to look at the organizational principles, again, of a region in the dorsal visual stream. And for that, we wanted to first see if there's a more coarse representation, which is called a contralateral preference, which means that information that is presented in one half of the visual field is processed in the other side of the brain and vice versa. And we also wanted to see if there may be even a more fine-grained representation in these areas related to attention. Now, regarding the first point, what we saw is that indeed there's this contralateral preference. So information on the left is processed in the right hemisphere and vice versa. And that holds for all of the experiments we tested. So that's a very coarse representation of visual space. Regarding the second point, we did not see a more fine-grained representation because the neural responses that we measured were fairly insensitive to the different positions that stimuli were taking in our visual field. But nevertheless, the experimental basis that we defined here may be very suitable for doing attentional mapping in the future. Now, in conclusion, throughout these 3 chapters, we looked at the topic of visual saliency from the 2 angles of a computational and a neuroscientific perspective, where we first identified computational factors that may explain saliency prediction, relating then computational models to the brain to understand saliency representations in the brain, and lastly, trying to find a spatial organization in regions in the dorsal visual stream that are very much involved in attentional processing. So I hope to have shown you that it can be very useful to take this bidisciplinary approach to studying visual saliency, and that this holds great promise also for future work on visual saliency. Thank you for your attention, and I give the word back to the prorector.
Thanks a lot for the very clear and nice presentation. The opposition will now be opened by Professor Alexander Zak, professor at the Department of Cognitive Neuroscience, Faculty of Psychology and Neuroscience in Maastricht.
Thank you, Victor. Dear candidate, first of all, my compliments and congratulations for indeed a very clear and calm presentation. It's very nice to follow. And also compliments for the very well-written thesis. It was a pleasure reading it. Very good use of scientific writing and very clear and well-structured experiments. And this compliments, of course, I would like to extend to your supervisor team. I have now approximately eight minutes left to challenge you on some of the things that you propose or investigated. And I would like to start on a very general level. And this is that I don't know much about computational modeling, but I do know a bit about spatial attention control. And what I was wondering when I read your entire thesis with the methodological approach, both on the computational but also on the neuroscience side, was to identify some sort of saliency computing region or saliency map. So everything was focused on the location in the brain. But in spatial attention control, we know that a lot of processing and underlying mechanisms are represented in the temporal domain with sampling rates of a certain frequency for more salient locations than others, probably represented in oscillatory mechanisms that play a huge role in understanding those principles of spatial attention control. So I was just wondering why you, if I understood it correctly, entirely neglected that part of the phenomenon of spatial attention control, and doesn't that limit your model? And is there a way to include such information also in a computational model approach on spatial attention? That's my first question.
Highly esteemed opponent, thank you for your compliments and questions. Indeed, we entirely discarded any temporal information, both in the eye movements, because we just look at fixation patterns as a whole. So just over also a fixed viewing duration. And indeed also with neural data, we look at fMRI, and we do not really have this high temporal resolution. So indeed, we are entirely neglecting that component. We still thought that it's informative to look at a distribution of fixation patterns in a scene, because they might just tell us what is the feature basis of where someone fixates and what is the underlying neural representation of this feature basis. So maybe our focus was more on features that are present also in natural scenes. So our entire focus was really on natural scenes and naturalistic scenarios, which is still not really solved, because indeed it's just for a fixed viewing duration that we look at a natural image, and we do not look at a sequence of fixations that are made over it. For that, we could still develop new models that take into consideration the scan path sequence. That's also something that is currently done more and more. And I think once we have models that are able to also take into consideration the fixation history to make predictions of new fixation locations, I think then we can also align it much better with more time-resolved neural data. You know, but the two have to go hand in hand, the modeling and the acquisition of neural data.
Perfect. Thank you very much. I'm really happy with that answer. If you allow, I would like to have a second, more general question. For this, maybe one of the paronyms could read out proposition 1.
Deep neural networks are an excellent tool for generating novel hypotheses about brain computations, but must go hand in hand with careful empirical validation of their predictions.
Yes, thank you. That sounds very logical, and my question could be completely stupid, but I was just wondering, I'm not using much computational modeling myself, but I was when I read this, I was thinking, well, if you can only if you always can empirically validate your models, that means you can measure them with the techniques we have available nowadays. And isn't that a bit, wouldn't it be more inspirational and challenging and interesting maybe to use the computational modeling part to fill the gaps where we can't yet measure processes on a different level, that helps us better to let's, for example, link the different scales of descriptions where we are stuck at the moment in many cases. Do you know what I mean? So if you can already empirically validate it, it means it's available for our way of describing in terms of space and time these neural processes, but maybe the interesting stuff happens beyond that.
Yeah. I'm not entirely sure if I understood your question, but correct me if I interpreted it wrong. So to me, the big benefit of these computational models is indeed to fill in these gaps that maybe neuroscientific experiments couldn't yet explain by exposing our networks to these possibly new scenarios that haven't been empirically done yet, and therefore being able to generate new hypotheses about brain functions. But at the very end of it, all these hypotheses have to be validated to really confirm if they have any use or not. So in that sense, I think the computational models really can pave the way for new directions of empirical research, but all these hypotheses are sort of worthless if they do not turn out to be also valid in the brain. They are more of an inspiration of where we can go to next with our experiments.
I fully understand your question. I indeed meant something else, but maybe I completely misunderstood the concept of computational modeling. And we can maybe talk about this in a different context, because I would like to use my last minute to ask a question that personally interests me the most. And this was in your impact section. You also talked about hemispatial neglect as a potential application of computational modeling. And you, I quote, say here that this digital twin of the brain concept, constrained by real-life observation, can be used to adapt interventions and thereby inform future developments aimed at functional neurorehabilitation. As a digital twin, I guess you mean the computational modeling of a virtual lesion in one of the two hemispheres, for example, in the frontoparietal network. And as you may know, I use quite a lot of non-invasive brain stimulation techniques where we do exactly the same thing. So we use NIPS in healthy volunteers to mimic neglect-like symptoms by temporarily inhibiting left or right posterior parietal cortex, for example. And then we can mimic those neglect symptoms, and we can test different brain stimulation treatment protocols to see how we can address those. What would your proposed model add to that, what we already do? Like how could that even be used together to get to even better diagnostic or therapeutic tool?
Is that we don't have the costs involved in experimentation, and that we can make a more large-scale experimentation of different scenarios and how they may influence then certain scenarios, certain outcomes and behavioral measures that we can then see in the very end. So I think it's just the benefit of the scale in this case, and that we can make it cheaper, we can make it bigger, and we can cover scenarios that may not be currently possible also when we just look at experiments that also try to make us understand better these mechanisms. But that very much requires that our models are tightly aligned with neural processing. And I think that's still something that has to be figured out, that there is this correspondence, that indeed a lesion in the model leads to similar behavioral responses as a lesion in the human would. Yeah.
That's great. So maybe you can validate your lesion approach with real lesion data or TMS-induced lesion data, and if valid, you could then scale it, like you mentioned, and still inform new protocols for non-invasive brain stimulation for neurorehabilitation. So that's very exciting. So I think my time is up. So I would like to thank you very much for your answers and give the word back to the prorector.
Thank you very much. The opposition will now be continued by Professor Bertrand Thirion at the Université Paris-Saclay in France.
Hello, I hope you can hear me well. Thank you for the very clear presentation that you gave, which was very well mastered, and it represents a huge amount of work. I was quite impressed by the amount of work that this represents and that was condensed here. Now I'm going to ask a few questions to better understand the implications of your work. My first question will be about chapter 4, about the topographic mapping of spatial attention in frontoparietal cortices. You mentioned in the document the difficulty of mapping effectively the PRF, the population receptive fields. I would like to hear more about how you diagnose this difficulty, because you took the best data that are available to the community at the moment. And so I'm wondering, or you generated some of this data. So I would like to hear more about how you see the difficulty. Is it a problem of quality, quantity, amount of data, complexity of the protocol? What could have gone wrong here?
Highly esteemed opponent, thank you for your compliments and questions. Yeah, in chapter 4 we encountered many difficulties with PRF mapping, and we attribute most of these difficulties to problems in mapping large receptive field sizes, because what we observed during our PRF mapping primarily is that in the search space of our receptive field sizes, many of the chosen values in the end were small receptive fields, although we know from previous studies that the receptive fields in higher order dorsal regions have quite large receptive fields. So that was a bit surprising, and that made us also dig deeper a bit into the reasons for this, looking then at these correlation landscapes when it comes to the optimization function of our receptive field modeling approach, which showed that there's just not much variation in the performance when it came to different receptive field sizes. So that shows us that noise may influence the estimation of receptive field sizes the most. That was really the main issue that we encountered, and we attribute this issue to the fact that the presentation screen that we used in our experimental setup was fairly small. So that means the distance between stimulus positions was not very big, and therefore our neural signal modulation was just not large enough to disambiguate between different stimulus positions. In terms of the complexity of the experiment, we were a bit afraid that maybe there may be some decline in attentional performance throughout the experiment, because we also asked a lot of our participants. We had 6 attentional runs that were each very challenging. But what we saw is that the performance was at a level that we would expect throughout the entire scanning session. So that showed us that people were able to perform the attentional task as we had hoped for. And therefore, we conclude that maybe this was at least not the main issue for the unsuccessful receptive field mapping in these regions.
So thank you. So at the end, would you recommend, for instance, doing more runs of these experiments to get a better SNR, or I don't know, sampling more subjects? What would make this more successful?
So in terms of relevance, I think the most relevant change would have to be to have a bigger presentation screen. It's as simple as that, so we can really cover more of the visual field. Besides that, I think if we would just repeat certain runs, I think that could potentially also help, because we also saw that there are quite some correlations between the experiments we chose. So for example, these flickering checkerboard bars and the bars that contained a certain motion direction, they gave us results that were fairly similar. So that means we could probably replace one for the other. The original idea was just to benchmark different experiments to see which one works best. But since two of those, but also others, seem to give us very similar results, we may replace the variety of experiments more in favor of repeating one experiment more time, which would give us a better signal-to-noise ratio and hopefully would lead to better estimations.
I see. That makes sense. Another question now is, I think in chapter 3, if I remember correctly, you present some receptive fields in polar coordinates. And you often present, I think you always present group averages of this receptive fields position. We could have expected to see individual ones. So why this choice? Can you comment on the difference in terms of quality, biases, etc., between the individual and group level maps?
Sorry, could you rephrase the question, please?
So could you comment on the differences you have between individual receptive field maps, so the map that represent the eccentricity and polar angle of the receptive field? So you present group level maps, okay? So why so? Why not presenting individual maps? So is there a reason in terms of quality of the results, or you noticed limited differences between subjects? So can you comment on this?
I assume you refer to the receptive field mapping results of saliency maps in chapter 3.
Yes.
Yeah, okay.
Absolutely.
So what we did see in individuals is just fairly consistent across individuals. So we see that in early visual regions, the mapping is quite successful, so that we can get these polar angle and eccentricity gradients as we would expect from dedicated receptive field mapping runs in natural scenes dataset, for instance. And since then it diverged a bit in higher order regions. We still saw that these mapping results were not maybe as expected, but these biases that we observe in the ventral and the parietal region of interest was also very consistent across individuals. And we thought it might just be a nice summary of our results to look at the group average here that may have more explanatory power in the very end when it comes to a bigger population. But the individual results were fairly consistent to each other in cases where we had some sort of trend. In some areas, there was not much to see, and this noise persisted. Yeah.
Okay, thank you. I think I have time for a small question. I don't see the committee. Do I?
Very small question, yes.
Okay, thank you. In chapter 3, you mentioned, when you talk about the banded ridge regression, that you run the analysis on a subset of 1,000 unique images to reduce the computational load of this approach. And here I was a bit amazed, a bit surprised. So what is the computational load? Is it in running the banded ridge regression, or is it something else, or the whole network, the whole analysis? What was costly there?
Yeah, so the computational complexity really came from the banded ridge regression approach, because there has to be an optimization of the ridge penalty parameter for each feature space that we had. And in the case of PRF mapping, we had, I believe it was 8 feature spaces. So already there was quite the explosion of computational power needed to do this computation. So we resorted to just using fewer instances to run this analysis on. Yeah.
Okay, thank you.
Okay, thanks. Thanks a lot. So the opposition will now be continued by Professor Tim Kietzmann at the Institute of Cognitive Neuroscience at Osnabrück University in Germany.
Thank you very much. I hope you guys can hear me all right. Great. So dear candidate, thank you also from my side for a well-written thesis and a great presentation of your work. I will dive right in to the questions because I have many. So we'll see how far I do with the time, but we can meet afterwards and we can discuss and nerd out on all the details. My first question is about chapter 3, where specifically you use NSD as a data set to search for signatures of a saliency map in the brain. And you do so by contrasting two models, the MSI model, which is the model from chapter 2, the ASPP modular model optimized on predicting saliency maps, versus VGG16, which is a model trained to categorize images. And there's a few causal claims in that chapter saying that because the saliency model outperforms the category model, that it has to do with saliency. And I want to, in being an opponent, I want to push back on that a little bit. First on the causal claims, but that's an obvious one, but maybe a bit more intricate. There are certain image statistics in optimizing for eye movements. And the point is that people look at faces a lot, for instance, right? Just as an example. So if you look at a model that's trained on ImageNet, ILS VRC, there's not many people in there and there's no categories for people. So if anything, the models use faces as like a side effect of categorizing a certain object. So faces are not important. But if you train on your MSI model on predicting eye movements, faces are central to predicting where people look. So I'm wondering if you find that in the higher level visual regions, including the fusiform, or also mid-level regions, which include OFA and such, whether you don't find the benefits of MSI because really it's more focused on faces than the category model. So that would be my first question.
Highly esteemed opponent, thank you for your compliments and questions. I think that's a very good point, and I also have to say that it's a possible confound in the results we see here. Indeed, in the model that is trained on the ImageNet dataset, I believe categories about humans or man, woman, whatever, I think they're not explicitly a task in the ImageNet data. It's not a class, it's not a specific class. I think it occurs more in the context of, I think, clothing items I saw, or certain sports, that people appear in these images, but it's indeed not the target of the network to then also learn what a face is, because it doesn't need to categorize it according to that. So it's very true that a model that is trained on saliency implicitly then learns to pick up on faces because they are attentionally very relevant. What I think should be done, if I were to modify maybe my experiments, is to maybe use as a baseline a model that has been trained on, for example, EcoSet that you're very familiar with, which contains categories that are more aligned with human representations, faces, bodies, and so on, which would give rise to representations in the network that are also sensitive to that. And I think if then the saliency model would still outperform this baseline, I think only then we can really robustly conclude that there's something more than just face representations and body features or something like that.
Mm-hmm. Thank you. I did think, to be clear, I did think that the autoencoder or image reconstruction objective was a smart move in that regard to build on the same image statistics, but I have a technical quibble with using MSE rather than a perceptual loss, but this is a detail. But it is doable, I agree. The second question, same chapter, is that if you're out to search for a region that is hosting a saliency map-like representation, right? If that were the task, then I was wondering whether you have the right tools in hand. So for instance, if you look at NSD, you have two hemispheres, left hemisphere being selected for the right visual hemifield, whereas your VGG and MSI models are fed the whole stimulus at once, right? So there are these technical details with comparing the models and the brain that may actually come in important if you're searching for a saliency map. And the point is that the regions that you're looking at in the brain do not cover the whole space, but the model does, right? And so you could make a similar argument for within a region, right? Where if you were to sample predominantly from the upper hemifield, let's say by virtue of your SNR or so, and then compare it to a saliency map of the whole image, could you really find what you're looking for? Like, or are there technical issues? The same goes for encoding models. We could make the same argument, but I was a bit worried, like, are we really having the right tools to search what we're searching for?
That's a good question. I think one part of the answer would be that we see fairly good predictions which show on some level at least a validation of the approach we took, because we can explain neural activity to some degree. Also if we consider the noise ceilings in our data, for example. But it's true that I think it holds great promise to look at models that are biologically more plausible, have even two hemispheres, have only parts of the input image be covered and processed differently in not just two hemispheres, but maybe even two visual streams, a ventral and a dorsal visual stream that may then get also different kinds of information. And by looking at models that have also interactions between these two visual streams, so that we get a model that is more aligned with brain data. We still thought that it's meaningful to maybe take these saliency models as they are currently, just to also test their limits and see if they have any validity as a brain model. Because also when we mapped individual layers to our brain, we did not see such a neat progression of preferred layers as maybe classification models have previously shown. So that shows us that maybe there's also differences to how the brain processes visual information and how the saliency model processes visual information. But I think it's a good first step to see also the limits of a saliency model and maybe take the results we obtain to refine our computational models of saliency and see if this refinement leads to then a better predictive accuracy of our neural responses.
Thank you, thank you. Final question, although maybe I'll sneak in another small technical bit. So final question is, if I'm sympathetic to the idea of modeling saliency maps, but I have, now it could be that we're barking up the entirely wrong tree, that we do not actually have an explicit topographic saliency map in the brain because, you know, as you're well aware, we sample information, and where we sample and how we sample depends on what we've sampled before. And in your thesis, you make this perhaps bold statement that saliency is a meaningful task in itself. And I want to push you back on that a little bit, which is perhaps surprising coming from me. But what would be an ecologically meaningful task? Like, I don't think we exist to compute saliency. Like, there are other things we do as to why saliency may be a good proxy for understanding what we're actually doing. But I wanted you to speculate a little bit on what these ecologically meaningful things could be and whether saliency map as a concept or whether we need a more, you know, embodied or active system to study this process.
I agree that probably saliency prediction is not the final goal of a visual system or a goal at all. I think maybe what I meant more is that training a model on saliency prediction gives us representations that have some validity in how they map to the brain. Maybe similar to how classification models showed benefit in explaining the brain, but I think we can probably agree on that the brain is not tuned towards image classification, but that also image classification is maybe embedded in higher order tasks, so that image classification may just subserve these higher order goals, let's say, of an organism. And I think for future work, it would be good maybe to not have a model that predicts saliency explicitly, but more implicitly. So really have some algorithm that does active visual sampling, where saliency may emerge as the optimal solution for solving a certain task. That brings a whole new set of questions, like what is then the task that we should train a network on, where saliency emerges as the optimal solution to? And I think there's also a lot of work going into that direction. And I think we can always take the next step of defining even higher order tasks that a lower order task may be the basis of. Saliency may be more a primitive not necessarily goal, but a more primitive task to train a network onto a line with the brain. But something maybe like scene description, scene understanding, I think is more promising as an overarching goal where saliency is part of the solution. May emerge, maybe not necessarily as a saliency map, but where the fixations at least show a similar fixation pattern, if we look at a certain viewing duration and so on. Yeah.
Thank you very much. This was very insightful. I'll return to the prorector. Thank you.
Thanks a lot. So now the opposition will be continued by Dr. Giancarlo Valente at the Cognitive Neuroscience Department, FPN in Maastricht.
Thank you, Prorector. So, dear candidate, dear Alex, I would like to join the other members of this committee in congratulating you with this work. And I would like to extend the congratulations also to your supervisor's team. This was a really nice thesis to read. I really enjoyed it, and I'm very happy I was given the opportunity to discuss it together with you. I learned a lot from it, and I think I like a lot this combination of computational modeling, but also advanced neuroimaging approaches. And I think this is the way to go if we want to get really deep insights in the problems. But I'm not here only to praise you, but I'm also here to ask you some questions. And I would like to focus a little bit on the statistical assessment, and on page 59. And I'm focusing now on the encoding models, and I'm looking at the paragraph number 2. I'm just reading now the first part, so to put everyone on track. So we are saying here that significantly predictive voxels were determined with respect to a non-parametric permutation test for each model, subject and ROI. Here we randomly shuffled the conditions of the true beta values, calculated the correlation coefficients with the predicted beta values, and repeated this procedure 1,000 times to obtain a null distribution. So what I would like to ask you, maybe I didn't get it from the text, but what I would like to ask you, did you retrain your, because this was obtaining cross-validation, right? So did you retrain your model for every shuffling of the labels, or was this done only in the test set when you compared a shuffled prediction with the true prediction?
Esteemed opponent, thank you for your compliments and questions. Indeed, we didn't retrain it because that would have been computationally very prohibitive in this case. We instead just shuffled the targets basically to obtain the statistic.
Right, so and I would like to question a little bit this procedure, because it can be shown that if you do use a cross-validation model and you do not perform this retraining, but you just shuffle the test set, this can lead to an increase in the false positive rate, right? So basically your distribution is, your null distribution is sharper than it ought to be should you do the things. But I mean, I understand that this was not feasible. And maybe then I would like to move also to the paragraph that just follows, which is the one of the model advantage maps, which I found also very interesting. And here I'm also would like to know, I'm just reading the relevant paragraph. So here we say that to that end, we first calculated the advantage of a given model based on correlation score for voxels that are significantly predicted by at least one of the two models. And then a null distribution was formed after randomly permuting the correlation score from the two models for each voxel individually and recomputing and so on and so on. So do I take it then that here every voxel was permuted individually, and then for each voxel, let's say you got a new preference for one of the two models, right? So maybe let's do a thought experiment. So I toss a coin 1,000 times, and you get 600 times heads. So is this convincing that the coin is biased? Do you think this tells you that the coin is not really giving heads half of the times, or maybe a bit more?
Well, regarding your question, I would say no, it's not necessarily convincing.
Okay, let's do it 10,000 times, and you get 6,000 heads instead of 5,000. 5,000, right? So you get more heads than tails. So in principle, the coin is biased. You should not bet with it. And the more samples you have, the more this is convincing. The problem here that I see is that if you permute all the voxels individually, there is a risk that you ignore the correlations between the different voxels. So what you think is, let's say, 1,000 voxels or for an area, or 2,000 voxels. Maybe effectively they are fewer because of correlation. Yeah, so what I will say is that also here the null, I think, is a bit too optimistic compared to the one that you will do. So here, maybe like if you were to look into it again, maybe you could consider other strategies. So what kind of approach could you think of to account for the correlation between the different voxels?
Honestly, I wouldn't exactly know what other strategy to use. If there's a way to decorrelate the individual voxel activations from each other and rerun this might be a feasible solution. I don't know if that's possible.
I don't know, to be honest. I mean, one idea could be, I wanted more to know from you, one idea could be, for instance, to scramble all the voxels at the same time. You have to use the same permutation for all the voxels, and then for each permutation you compute a prevalence map. So if many voxels are correlated, if they prefer one condition, they would also all move to the same preference, and then your null will be a bit larger. But I mean, that's okay. I'm just a little bit nitpicking here. So if there is time for another question, I will ask you one more. Otherwise, I will give a very small question. One thing I was wondering, the results are great. If I look at figure 3.4, you see that basically everything is significant. Which is good, it's fantastic, but usually this depends on who you are as a person. I'm a very, let's say, not optimistic person, put it that way. And when I see lots of positive results, I always worry that there could be something like some bias lurking or something like this. And this relates a little bit to question 1, when you only permute the test labels without permuting also the training. So could you, let's say, are you sure that your null distribution, or let's say the null of your encoding model, is exactly 0? Because what you're doing here is flipping with respect to 0.
Yeah. Yeah, I don't know exactly how to address this. I think, indeed, because of the points that you are pointing out, maybe our results are a bit optimistic. May I just ask which significance results you refer to?
So I'm looking at panel A, for instance, in the encoding. Figure 3.4, panel A.
Ah, okay.
So I'm seeing that in all the areas that you consider from the early up to the parietal. So I'm not questioning your results. I think there is a nice modulation and it fits very nice. I just want to think together with you whether you trust that all the values, for instance, also the ones that you get in ventral and parietal, are actually significant.
Yeah.
And
yeah, I mean, the statistical tests we chose at least have shown us this significance when compared to a 0 baseline. Indeed, in the ventral stream, especially for the low-level categories, it was fairly close to the 0, although we did not observe much variation. So also the standard deviation was very low. So in a sense, maybe we did not specifically distrust these results, but yeah, we probably should have consulted you for better statistical validation.
Okay, thank you, Alex. I'm very happy with your answers. Thank you.
Okay, thanks a lot. Then now the opposition will be continued by Professor Bernadette Jansma, professor at the Cognitive Neuroscience Department, FPN, in Maastricht.
Thank you, Prorector. The candidate, first of all, also let me congratulate you. I enjoyed reading your book immensely. I learned a lot. I enjoy also following the interdisciplinary collaboration between psychology, computational neuroscience, and data science is also a nice test case of cross-faculty collaborations and wonderful work. But I was a bit surprised about this idea of saliency and the relevance of saliency in general for human perception and object identification. But okay, so I read a bit more critical, but in chapter 2, for example, you clearly said, when I incorporate, I'm more positive than my colleague. So obviously if I add ASPP into a model, the performance is better. So and then I said, cool. That means when the network knows a bit more, eventually top-down by learning, it is better in producing the salience map, which indirectly is a measure of predicting visual gaze control in humans. So my question then is, what is this ASPP? You say it includes global context based on semantic features. And that's quite a lot to say in one sentence. So what do you mean when you say global context? Or what do you mean when you say semantic features?
Highly esteemed opponent, thank you for your compliments and questions. So this ASPP module, what it does is that it operates on top of very high-level feature representation. So it operates on top of the output of our encoder, which is based on this classification network. So it should have a notion of more semantic concepts and representations in a scene. So that's where the part comes from, that it's sort of semantically informed. The global integration of information comes from the fact that this ASPP module has layers with differently sized receptive fields. So it also takes into account very large receptive fields, and in the extreme, even a receptive field that covers the whole feature space. So it really summarizes an entire feature map by just one value, in fact, which we relate to, and which has some parallels to the scene gist idea, which was originally based on low-level features, but the mechanism was similar, that just a low-level feature map was also summarized by just a single value.
Yeah, and that brings me to another question. So obviously then the network is wise. It has learned, for example, you gave also examples of pictures like the elephant. There was a picture of the elephant, which triggered my train of thoughts and say, okay, so obviously you are able to provide very nice salience maps on this picture of the elephant. You also see nicely that actually hotspots are in the eye and around the tooth, which evolutionary also makes sense. So I was very excited to see that. And then I thought, okay, so in language we, by the way, call this saliency prominence, and we have that at different levels. And I can also modulate prominence or saliency, for example, by situational context. So if I have the sentence, yesterday I went to the zoo and I saw the elephant, versus yesterday I went to the supermarket and I saw the elephant. Would you then actually think that your saliency map would be identical of the elephant in these two cases? And if yes, why?
In the visual domain only, you mean?
Well, that's already a good question. So why are you asking this question?
Okay. So, in my opinion, this ASPP module as is not necessarily able to also capture these very high order contextual differences. That's actually something that still needs to be tested, because there are also data sets that have these object scene inconsistencies. Also, like you say, maybe an elephant in a context that you would not expect it. This might, though, require a more high-level understanding of what is normal in our visual world, at least. And therefore, if there are any violations of this prediction of what is normal, that this may be then considered salient.
Yeah, and that brings me, so it's clear you already show that you understand what I'm up to. But it's clear I also sort of miss a bit on overall theoretical framework. And I just, of course, came with my favorite one, which is by Foster, which is the perception action cycle. So what are you working at? You try to understand how we perceive and interpret the world, right? And you, of course, want to integrate this in a model. It's also nice. But when you then think about the Foster story, that actually whatever we see comes into the cortex, then there are some hypotheses generated about what we see. Hippocampus basically check with what we know, and whenever there is a match, they say, yeah, we don't need much neural resources because we fully understand the situation we are in. If there's a non-match, there's uncertainty in the system, there's a increase in remapping, rematching, rechecking what I see. So you already also clearly say that you cannot do temporal resolution in this, but do you have any idea how you could incorporate basically several of this? I just call it the ASPP filters. So you have filters on visual features, semantic features, emotional features, because I can have the elephant in a nice cuddling context. I can have the elephant in a context where I'm attacked by the elephant, right? So do you envision for the future another architecture of your network in that sense?
Yeah, absolutely. So I think there are still many open questions and many shortcomings of the current models, because indeed it doesn't consider emotional state, it doesn't consider even temporal aspects or fixation history, which would inform future fixations. So I think one way to go for new models would indeed be to equip them with more higher level understanding of a scene, which could include also emotional aspects of a stimulus we look at, because also it has been shown that maybe a face that displays a certain emotion attracts our attention more than one that is maybe more neutral. So I think that's one way to go, that we really try to bring in even more components together that also have interactions between them to make a more holistic prediction of what is salient. In the other direction, we can also make improvements, such as adding low-level components that also allow us to work well with examples of low-level feature pop-out, because that's also something that the network cannot perfectly solve yet. So I think there are ideas for many directions and how to improve models in that regard.
Thank you. Yeah.
Thank you.
Alexander Kroner, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request you and your company to await the results of our deliberations and our return in this room.
All right.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
The PhD defense has now ended. The degree committee will debate.
There it is.
Please be seated.
Alexander Kroner, the degree committee here present has assessed the quality of your thesis and your defense, and in view of its positive verdict and in taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Goebel is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Alexander Kroner, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Before I hand over to Mario, who gives the deserved laudatio, I just could not give it over to him without saying that I want to in person tell you how much I enjoyed working with you for quite some years. Mario goes more in details, I guess. But I also want to congratulate your partner and your family. Sie können heute sehr stolz sein auf den Alex. And I also want to say that hearing you speak, it reminded me how you are. Smart, gentle, modest, very modest. You know, your work I used a lot in the Human Brain Project. It's cited more than 200 times after a few years. You know, it was in robots, it was embodied. You mentioned these things not because you're just too modest, which is wonderful. Keep it. But also learn to say when you have to say things, right? And I think what we also all acknowledge is that you know where to say, this I don't know, which is wonderful. It's a strength and a maturity of an academic person, which you are. Yeah, I want not to say more than that. I want to give over to Mario to give the laudatio.
Thank you, Rainer. Alex, also of course, welcome everyone. Hello, dear friends, colleagues, also the family of Alex. In contrast to Giancarlo, I'm actually allowed to only say nice things about you and just praise you all day long, so that's kind of a fun thing. So I want to start with, I'm also very happy with what you did today. You're such a brilliant young scientist, and really how you managed this, how you had this presentation, and how you handled the questions, it was truly amazing. It was very, very, very nice to see that. I'm very proud of you. But of course, we also have a longer history already together. So the first time I met Alex was when he actually came as a master student, right? You were looking for someone to supervise your master thesis, and you ended up with us. And we are very happy that you came to us, and because we were very impressed with your enthusiasm, with your technical skills, with your deep interest in saliency. So of course, for us, it was quite clear that you would become a PhD candidate. even though for you, that made a little bit, meant a little bit of a shift, right? A little bit going out of your comfort zone from nice AI, that was where your heart was, that was what you really knew, and then suddenly you end up in this world of cognitive neuroscience and all these strange things. But of course, you first, inside your PhD, remained a little bit in your comfort zone, stayed with the AI, and you built this very nice model that we have seen here today. And I think what was not mentioned so much, but what I thought is very cool, is the impact that this model had. It was very well received in the scientific community and used by many other people. But it also went far beyond that. And I think maybe the best example is when an artist contacted you and actually used this model for pieces of art where he identified what is very salient in an art piece and then either exaggerated this or removed this. For those of you who haven't seen it, it's actually, some examples are in Alex's thesis. There's just one, the one with Trump, that still gives me nightmares. Maybe try not to look at that one. It's spooky. Of course, after leaving your mark on art, neuroscience was not a big deal anymore, right? Now you were ready. And then you started even to do your own fMRI experiments. And you had a very first, very ambitious project, right? And maybe it was also a little bit the prospect of torturing me here and there that you took on this project, because for those of you who don't know, Alex put me in the scanner for several hours, 10 days in a row, and I had to look at many, many, many images. Unfortunately, that project did actually not end quite the way we were intending it and what we had hoped for, which is, of course, also something that is that really showed your character, right? A PhD is not always smooth sailing, and you have had your setbacks with this project, but also with the last chapter, where in the end, even though the stimuli were very nice designed, maybe the screen was not big enough and we did not have the intended results. But what impressed me very much is how you were able to not only work through these promises, but basically how you use this as a stepping stone to, in the end, actually produce a very, very nice thesis. And that's something that not everybody can do, and not everybody has this kind of perseverance. And I do think this kind of perseverance for you really comes from the fact that you are, in general, from my experience with you and our interactions, a very well-rounded person. So Alex is not just a brilliant scientist, but also a very valued friend of many people in the lab. And seeing you and Marta together, he seems to be also a very great partner. You have a particular fondness for hip-hop that we share. Kendrick Lamar. You're not just pimping butterflies, you're also pimping terminals, I know. You're a cat lover. And a culinary experimenter, right? You make very delicious pancakes. And I heard once in a while you also bring some Romanian moonshine that has caught on and has become popular. Maybe, like Giancarlo, I have to say one negative thing. You don't have a particularly good sense of direction. And that is something that we share. So I remember when we were going in Glasgow to print a poster last minute, because we still had to get this one, and we almost got lost in the city, and we were very happy that we actually made it back eventually to the venue. Don't worry, that's the only negative thing I have about you. Well, Alex, to sum it up, your dedication, your innovative and creative thinking, your perseverance in the face of challenges, they are really inspiring. And as you move forward in your career, I really have no doubt that you will continue to push boundaries, also in your new lab. And I think you, your family, and Marta can be very proud. Sie können wirklich stolz auf ihn sein. So, congratulations, Dr. Alex. May your future endeavors be as successful and enriching as your time here has been. And remember, should you ever get lost on your way to a conference or a presentation, you can always count on me on getting lost with you.
Dear Dr. Kroner, also on behalf of Maastricht University and of course of our Faculty of Psychology and Neuroscience and the Department of Cognitive Neuroscience, I congratulate you with the degree you acquired. And I also, of course, congratulate the supervisory team and Marta, of course. Und herzlichen Glückwunsch auch an die Familien. Sie können wirklich sehr stolz auf Alex sein, oder eigentlich sollte ich jetzt sagen Dr. Kroner. Thanks also to the members of the committee present online and on site. Thanks a lot for the lively and very interesting discussions. And actually, then I would like to invite the audience, because now we basically close the ceremony. I would like to invite the audience to already go to the reception at the Refter, and we will then take here still some pictures with the family and with the committee. So, hereby I close this academic ceremony.
Vielleicht kommst du irgendwann wieder zurück. Jetzt habe ich so viel Stille. Nach vorne?
In die Mitte.